start by really explaining a little bit of the background and why we're hosting this webinar series in the first place. Partners for Access, we're a specialised consultancy and we focus in on orphan drug access. And as part of that, more of our work recently has been involving gene therapy. And we've been struck by the level of interest, but also actually the level of confusion there is around the area globally. And today is really about responding to that interest and hopefully clearing up some of the confusion. You can go to the next, thank you. There's certainly no doubt that since the Human Genome Project began last century in 1990, that the focus, the attention, and also the hype around gene therapy has grown immeasurably. And some estimates, obviously they vary, but some estimates place gene therapy development as the second major R&D focus pre-launch. So clearly there's lots of resource being made from the pharmaceutical, the biotech area, lots of resource being made, and clearly significant returns on investment need to be reaped if it's going to be successful in the future. But obviously it's not, and I think everybody could probably agree that it's not necessarily been a truly successful journey so far. And Glybera and Strimvelis, two of the first major gene therapy launches, have widely been described as commercial failures. Now, there are several reasons for this, notwithstanding the size of the patient population, the practicality and pragmatism when it comes to the administration, the site of administration, and of course the cost of production, not to mention some of the gaps in some of the clinical evidence. So not necessarily being completely clear so far. However, here we are, and still there is a lot of attention and a lot of excitement around this area. And regardless of whether gene therapy is a revolution or an evolution, we believe at Partners for Access that we all have a role to play in developing a better understanding of the science, better understanding of the opportunities, and also, very importantly, a better understanding of some of the pitfalls to avoid. And for this reason, we're focusing our first webinar on dispelling some of the myths of gene therapy and certainly highlighting its potential. So what I would like to do now, we are making this an interactive session. What you should see on the right-hand side of your screen is an ability to be able to answer a poll. And we're going to give you 60 seconds. We've got 1 minute. There'll be 3 questions coming up throughout this session. And what we would like you to do is to answer the poll questions. So the first question is multiple choice question, obviously. And the first question is, how would you rate your knowledge level in gene therapy? So A is that you have a good level, or perhaps a very good level of knowledge in gene therapy. B is that knowledge is fair. Or C, which would be surprising if you're on the call, but C is that you haven't heard about gene therapy and certainly you are not very aware of the area. So if you can take a moment to answer that question, and then what we'll do throughout the session is we'll be able to respond to some of these questions, and certainly it'd be very interesting to understand everybody on the call today and everybody's understanding level, or at least how they think that they understand gene therapy. So let me see, we've got 5 seconds left, and we'll just see the poll results coming through. Okay, right, the poll has ended now, so hopefully everybody has voted. Let me see. My technical ability is relatively poor, so you have to bear with me as I go through here. So let me see, here we go, the results. So actually nobody, interestingly enough, Rena, you're going to be talking a bit about this, but nobody has rated their knowledge as good. 75% or about 3/4 of participants have rated their knowledge as fair, and interestingly enough, Just under a quarter of participants say that they really have a very poor knowledge of gene therapy, or perhaps they are not familiar with the area at all. So, Rena, I'm not sure what you think about that. Does that surprise you or not?
Actually, Sophie, it might surprise you to hear that I answered fair myself.
Oh, okay.
As you will note in my presentation coming up, there's a lot of confusion even from the scientific point of view. So I'm not at all surprised if people who are not even in the sciences would also be confused.
Excellent. Okay, well that's a great start and a great introduction for your presentation. So let's move on, and as I say, I gave you a very brief introduction there. I was fascinated by your talk that you gave at the World Orphan Drug Congress, and it would be wonderful for you to share from a scientific perspective the opportunities that you believe gene therapy has, and also some of its practical limitations as well. So thanks very much, Rena.
Absolutely. Thank you, Sophie, and thank you everyone for joining us today. It is my pleasure to provide a brief overview of the science behind gene therapy. And my goal really is to get us all on the same page and use this part of the program as a jump-off point for further discussion. So let's start by defining what gene therapy is. As Sophie mentioned, there is a lot of confusion in the field, and even the definition itself can be quite confusing because the term is used in many different ways by many different people in many different contexts. I'm trying to get to the next slide. Anyone else has control? Kindly change the slide for me. There you go. So what I'm showing here is only one definition, and this is a definition by the US FDA. So the US FDA defines gene therapy or human gene therapy as the administration of genetic material to modify or manipulate the expression of a gene product or to alter the biological properties of living cells. Now, there are two important elements to this definition. The first is that the therapeutic agent is composed of genetic material. The second is that the goal is to modify or manipulate gene expression to treat diseases. So indeed, genes are both the therapeutic agent and the therapeutic target. So how does gene therapy work? All right, so in normal cells, DNA, which is our genetic blueprint, encodes a message called RNA, which is then translated into a gene product called protein. Proteins serve different functions in the body to help us stay alive and healthy. So for example, proteins can serve as structural components or can perform enzymatic functions. What happens in the disease state? Genetic diseases are caused by defects or mutations in DNA, which leads to the encoding of a defective RNA, which in turn translates to a defective protein, or sometimes no protein at all. So for example, patients with a mutated LPL gene end up being deficient in LPL protein or LPL enzyme. These patients cannot break down fat and are restricted to a low-fat diet and suffer from recurrent life-threatening pancreatitis. So how can gene therapy help? With gene therapy, a normal functional copy of the DNA is introduced into cells, which then encode functional RNA and therefore functional protein. So this is exactly the case with Glybera, which is the first gene therapy approved in Europe. A normal functional gene for LPL is introduced into the cell, which then encodes a functional protein that compensates for the deficiency in these patients. Sounds simple enough, right? In theory, yes, but in practice, one of the major hurdles of gene therapy is getting the normal functional copy of a gene into cells to begin with. To accomplish this, scientists must employ gene carriers, of which there are two major types: viral vectors and non-viral vectors. Viral vectors can integrate DNA into the host genome, or can be non-integrating. Non-viral vectors can involve physical manipulation or chemical agents to carry DNA into cells. Each method has its own advantages and drawbacks. Let's take a closer look at viral vectors, as they are the more commonly used carriers. So viral vector approaches exploit the fact that viruses have evolved millions of years to be effective carriers of genetic material and effective infectious agents that readily enter human cells. For gene therapy, the nasty pathogenic parts of the virus are removed, and the remaining components are used as a package to carry a new gene into a cell. So note that integrating vectors are so named because they integrate genetic material into the host genome, such that they essentially become a permanent part of the genome. Now, while this has advantages for longer-lasting effects, integration can sometimes disrupt host genes, leading to unwanted effects such as the development of cancer. There are two ways by which viral or non-viral vectors can be delivered into humans. To the left on your slides, you can see the in vivo method, which involves direct administration of the therapy into humans, usually by directly injecting into the tissues involved in the disease. To the right is the ex vivo method, which refers to a method whereby cells are manipulated outside the human body. So cells are first harvested either from the patients themselves or from a matched donor. The gene therapy is then introduced into the cells outside the body, after which the cells are administered back into the patient. So now that we have the basics covered, let's address some myths or some frequently asked questions regarding gene therapy. So the first question I have here is, does gene therapy always change the gene? Well, the answer is actually no. It doesn't always change the gene because it depends on the approach. While gene editing approaches such as CRISPR-Cas9 involve changing host DNA, many other approaches do not in fact change host genes. I have some examples shown here on the right. So first, for example, most classical gene therapy approaches involve gene augmentation, a strategy whereby a healthy version of a gene is introduced into cells without getting rid of the abnormal gene. Another exception involves gene silencing, an approach used for diseases caused by the overproduction of the RNA message. So in this case, the approaches typically target the RNA message and not the DNA itself. A third exception are immune activation strategies, typically used in cancer. Here, the goal is to activate the body's immune system so that it can get rid of the cancerous cells on its own. So usually with these approaches, the genes that cause the cancer themselves remain untouched. So hopefully we've addressed that myth. Let's take a look at another myth or another frequently asked question, which is gene therapy only used once? Now, several gene therapies at this point have been shown to have long-lasting effects, suggesting that one-off therapy is certainly possible. But it's really hard to say at this point, as the field is young and long-term follow-up data are not yet available. The long-lasting effects of gene therapy depend on a few factors I've listed here. So first, it depends on the ability of cells to survive once they uptake the therapy. As many of us know, most cells in an organism eventually die. If these cells are not replaced by new cells, then the therapy dies with them, so to speak. Another consideration is that sometimes cells that contain new genetic material may be seen as foreign by the immune system, which may then reject the new cell. Finally, the transience or permanence of an effect may depend on additional engineering built into the therapy itself. It is possible, for example, to design therapies that contain a kill switch, meaning it allows us to turn off the gene therapy in case it leads to adverse events. Now, the question of permanence is also related to another potentially controversial topic in gene therapy, which is, somatic versus germline gene therapy. Somatic gene therapy is limited to the patient and is non-heritable. Germline gene therapy, on the other hand, targets reproductive cells, so the egg or the sperm, and can therefore be inherited and passed on to the gene pool. Now, while germline therapy is not allowed in Western countries, the ethics of germline therapy and of gene therapy in general is still a timely and important topic of discussion. And finally, to answer the main question posed for this webinar, is gene therapy an evolution or revolution? From a purely scientific point of view, my answer is that gene therapy is the result of an evolution of scientific progress. It is currently possible only because it stands on the shoulders of giants. As you can see, the idea of gene therapy began way back in the 70s. That was 4 decades ago. It came about because of the development of recombinant DNA technology, which, if you think about it, would not have been possible without the prior discovery of genes, of DNA, and of DNA structure. Since those days, other major advances have allowed the field to develop, including, for example, the discovery of CRISPR in bacterial genomes, the completion of human genome sequencing, and the evolution of next-generation sequencing methods. And it's not just the successes, the failures are also important for informing the development of the technology. In the last couple of years, we have started to see the fruits of this very long labor with the regulatory approval of several gene therapies. You can see towards the end of the timeline between 2015 and 2017, we've seen a lot of approval. So I'm going to end here for now, but I hope that I have provided everyone enough background, tools, vocabulary, and ideas to facilitate our discussion as we move forward with this webinar. Thank you.
Thanks, Rena. That was excellent. Thank you very much for that overview. And maybe, certainly we can open the poll up for questions. If anybody has any questions, they can submit them in the right-hand side of the screen. You'll see a Q&A section there. If you want to send through your questions, you can. And while we're waiting for some questions to come through, I have one specifically when you were talking before about the difference between in vivo and in vitro application, and also as well the scale that you just had there before, the impact, it certainly seems to be increasing at a very rapid rate recently. So earlier this month, there was the first person was actually given in vivo gene therapy as part, it was a Hunter syndrome patient. From your perspective, where do you think that the greatest value lies in gene therapy? Is it through the in vivo application or is it in vitro?
So actually, I was talking about in vivo versus ex vivo.
Yep.
And right now, there have been a lot of ex vivo approaches,
Yep.
Taking out the cells from a patient, right, and doing the manipulation outside the patient. And that's
Yep.
because of the nature of the diseases. For example, if hematopoietic cells are fairly easy enough for us to harvest at this point, and so we can harvest them and we can manipulate them outside the body and easily put them back, and they can grow into the type of cells we need them to become in order to be therapeutic. But that's not always the case. So some cells are harder to harvest. Some cells are harder to change into the type of cell we want them to be once they enter the body. On the other hand, for ex vivo therapy, you may necessarily not have to expose the body to the virus. Does that make sense? Because the therapy and the viral treatment is happening outside the body. We don't have to expose the patient to the virus directly, which can have implications for safety.
So, I mean, it seems to be sort of a trade-off between the practicality and the safety then of application.
Absolutely.
Is that fair? And where do you think that the future is heading, though, or do you believe it's very much specifically around the condition itself?
I think it will definitely, I think it will depend on the condition itself, whether it can be accessed in vivo or not.
Right.
It can also depend actually on manufacturing, because both manufacturing of viral vectors as well as trying to grow enough cells ex vivo to put back into a patient. Those kinds of things are not really all that easy at this point. So manufacturing might play a role in the decision to pursue either in vivo versus ex vivo methods. Keep in mind that in vivo methods can also sometimes be very difficult when we're trying to access specific tissues or cell types. Many of the gene therapies available today involve very involved procedures, actually, intramuscular injections, subretinal injections. So those are also not trivial in terms of clinical application. So we have a lot of things to improve upon in terms of manufacturing and delivery / administration. And I think whichever technology gets addressed first will probably move the path forward.
Sure. I mean, from what you're saying, it still sounds like we are very much still in the embryonic stages.
Correct.
Right.
And the manufacturing and the clinical development side.
Sure, and Amir will touch on this shortly. Just had another question come through in terms of, let me just read this out. What steps are taken to ensure that inserted genes are correctly targeted and do not lead to adverse events like cancer?
Right, so we're referring to the technologies that are integrated into the genome, which means they can potentially disrupt host genes and lead to cancer. So one approach is to, for example, use non-integrating vectors. That's one way. Obviously, because then we won't have to deal with integration. Things like CRISPR-Cas9 are now very precise ways to edit the genome, which means we don't need to randomly target the genome anymore. We can actually precisely hone into the target gene of interest and make only the changes in that site. So there are those things. Now, we're still, again, in embryonic stages with those technologies. So we still have to think about off-target effects and trying to improve the technology to minimize these off-target effects.
Okay, excellent, excellent. And I guess my final question, which is sort of links to what we've been talking about, is there one specific disease area that you believe is more appropriate for gene therapy?
I think at this point we might want to focus on monogenic diseases. Diseases that are caused by a mutation in a single gene. That way we can design a straightforward path pre-clinically and clinically. Things like neurological diseases or obesity or cardiovascular diseases, which can be multigenic, might be a little more difficult at this point to deal with because we're not going to be targeting only one gene. And we don't even really understand how the different genes that cause these diseases might work with each other.
Excellent. Thanks very much. And I know that Amir is going to touch on this too throughout his presentation. So thank you very much for that. That was great. If anybody else has got any questions as we go through the webinar, then please feel free to use the Q&A section on the right-hand side of your screen. So let's go to another poll question. So this is 2 out of 3 of our poll questions. Hopefully people are now familiar with what you need to do. So on the right-hand side of your screen, you're going to see shortly, you can see it now, 4 multiple choices coming up. And the question is, what you believe are the key challenges of gene therapy? So you've got 60 seconds to answer the question. A is the lack of clinical data. B is the lack of patients, C is the lack of access, or D is actually all of them. And we're going to very much enjoy the responses that came back. Certainly I was quite surprised with the other responses that came back, so I'll be very interested to see what people's answers are to this. I have an idea what they're going to be. But we'll see. And I believe we've got the, if we can see the answers coming through. Hopefully they'll come through now. Excellent. So, quite an interesting mix of different results. So we've the majority of people, just under half, 42%, believe it's all of the above, and perhaps even other things. Just over a quarter of you think it's the lack of clinical data, and certainly some of the early experiences with Glybera, we could probably say that was due to the clinical data, or at least looking at a slightly different patient population to the one that the company ended up focusing on. The lack of patients seems to be relatively low. 11% of people have said that, and then 16% saying lack of access. So it very much seems as if it's a combination of all of the above, with a focus on the clinical data. So, Emir, I'm not sure what your initial thoughts are to the answers there, whether you were surprised at that or not.
Hi, good evening, everyone. I am not actually surprised. I believe the people know much more than they think they know, and they just sometimes have to just make up their mind, and that's the result of it, in my opinion. But yeah, I think that's the short answer to the question. And yeah, I think it would be quite interesting to see after that we finish the discussions this evening, then, or in the morning in US, then what would be the outcome and how they perceive the gene therapy so far. After that, they have heard us speaking.
Sure, to see if any changes have been made.
Yeah,
okay.
any changes into, how can I say, into the environment, hopefully positive, and yeah.
Okay, excellent. All right, so without further ado, as I obviously gave you a brief introduction at the start, very delighted that you're able to join us today and to share some of your thoughts, really from a commercial perspective, obviously, looking at patient access, and also the ethics as well when it comes to gene therapy. So can you have control over the slide?
I'm trying to move it.
Okay.
Okay, it's moving. So
Perfect.
yeah. I think, hello everybody, good evening. Thank you for being here. It's a pleasure to give some information around the gene therapy. The scope of this talk is to, I'm sorry, I think the whole thing is not responding. Can somebody move the slides? Thank you. I would appreciate that. So continue. You'll hear me saying next slide and next slide. So the scope of the discussion is about giving you some examples of my experience in gene therapy and how that impacts into the areas of patient access and ethical perspective. The first element that takes part into this discussion is, how is the big picture formed and what are the biggest players? I think we see politics, we see regulatory, we see R&D, and we see the patient. At the end, there is an upper part which is all made of politics, where we know what politics should do. So they are there to help us to understand, basically to safeguard the public health, but also provide solutions, tangible solutions. And we have seen it in tangible solutions by creating organizations like inside EMA, having the CAT that is responsible for cell and gene therapy, where CAT has very good discussions and help the companies to develop to develop the gene therapy. And this is more an integrated partnership between the R&D and manufacturing part of the companies, and then with the aim to provide something to the patient that is tangible and that can address the unmet medical need. However, we see that in order to succeed the real picture, it's quite challenging. We see there is a need of integration of manufacturing of the manufacturing platform, the vector that is being used, the promoter that is being used, and the administration route. In addition to that, we see the examples that have arrived of gene therapy in the market. We see Glybera, which effectively worked into a patient. The problem was that the patients were too few, and then it took too much time to arrive into the market. This, without saying of what happened with the product itself. I think the real part of the Glibera was that essentially it should have been developed into a more integrated way and to provide and to address these problems. In addition to that, then we see Strimbelis, which hit now the approval, actually now last year, and actually it's a big hope for patients that will become might become quite cumbersome if the mother company doesn't find a way to address basically all the existing patients with this disease that are actually into this situation of that have the disease. And in the third part, we see a proliferation of approvals. We see a proliferation of approval that is based by, I think it's coming more from the real integration between the regulatory authorities that help the companies, that sit together with the companies and design basically a path to the approval. So help them to design the clinical studies, help them to talk to and also help them understand what are the real patient needs. So this integration of regulatory enterprise factors and the patient access will be crucial in the next run to design more and more gene therapy treatments that will be able to address the real and medical needs, and not only have 1, 2, or 3 patients being treated all over after the approval. And that would be the most important part. In the next slide, I would like to move on and give you the understanding of what are the Can you go up one slide? I think I moved too fast. Yeah, we are on now. The next slide is helping us to understand what are the elements for success in embedding gene therapy into the healthcare system. The primary focus is to develop the right infrastructure and to use the right infrastructure. Then, what does it mean? It means that to have access to adequate treatment centers, that is essential to succeed in gene therapy, especially in the clinical and not only clinical part. But you need to have access to patients, basically. You need to have access to people that convince the patients to go and get the therapy, and the patients and the whole organization, the healthcare workers need to be convinced that what you're providing is the right balance. It's providing the right benefit. In addition, in order to develop that right benefit, you need strong science. And here I have evidenced a number of elements that are related to the strong science. And after that, then you need to deliver in high quality related to that strong science. What does it mean? You need to have a proper technical development done properly at the highest level of R&D that we know so far. This is not an academic exercise. This is the cutting edge of all technologies put together, and as Rina explained. So into that part, we need to put together the best CMC science for process development and all the other elements that have to do with the value chain of the product, from starting drug substance, drug product, and final delivery of the product. These are essential, and we should never forget that the global development competencies are key to succeed in gene therapy. You need an executive leadership that has a vision for this kind of therapy. You need business strategy, and you need global market access associated to real effective access programs that are really making the difference. In the next slide, the next slide is reporting basically, can you go up one slide? Okay, yes. The next slide is reporting what and who determines the eligibility criteria for patients, and this is essentially one puzzle that is composed of 3 major category players: patients and healthcare workers, regulatory authorities and policy factors, and enterprise factors. I will expand to them into these 3 elements. The desires of these 3 players should be integrated to achieve the best treatment possible for patients. Without that, this is aimed to fail. The next slide describes exactly how these elements for success, what are the elements for success to embed the gene therapy into the healthcare system. So the infrastructure, sorry, what happened? Yeah. The infrastructure that we mentioned before and all the elements related to that, then it has to do with the patient impact, it has to do with the regulatory and guidelines, with the guidelines being driven into the regulatory area, but also with the regulatory and clinical aspects. But it has to do foremost with the R&D and portfolio strategy. So what does it mean? That once you define the grade of severity of the disease where you want to intervene, and once you define the viral vector, the gene, the correct mutation, and the working the correct mutation, the immunosuppression strategy if it is required, the product presentation, all these elements, they have to be tied up to the patient. They have to be tied up to the final patient. This is essential. In order to do that, you will see that the patient will have encountered the disease. They will have some neutralizing antibodies versus the capsid, versus the vector, versus the other viral elements. And they might have also other problems. So you need to cope with all that. And these will be the patient population that will be free from these elements. That will be the inclusion and exclusion criteria of these patients that will be going into the study. So into the next slide, I have provided you a stage gate approach of how to determine which patient will be possible and the most suitable to arrive to a treatment. The point is that as the diseases are orphan diseases for the moment, the diseases are also monogenic. Then when you start cutting the patient population in numbers, then the number of patients actually becomes quite very small. So in order to understand it,